MiDOG. I'm Lina. I'm the CEO of MiDOG Animal Diagnostics. And today's webinar is hosted by both MiDOG as well as the SCVMA, the Southern California Veterinary Medical Association. I'm very excited about today's talk on our collaborative study that led to the discovery of a novel Castellaniella species in a fatal canine infection. CE credit is available for this meeting today. You will see at the end of the presentation a QR code to download the certificate, and we will also put the PDF file in the chat of this webinar so you can access it right there as well. We'd love to hear your questions, and we will go over all of them at the end of the presentation. So please, if you have any questions, please put them in the Q&A chat. And with that, I get to introduce today's speakers. We have two fabulous speakers joining us today. The first one is Dr. Mark Yacoub. He's a scientist here at MiDOG Animal Diagnostics. And here he analyzes shotgun metagenomic data, and he leads our efforts to assemble and investigate the genomes of animal parasites. Dr. Yacoub earned his PhD in microbiology from the University of California in Riverside in 2024, and his bachelor degree in microbiology from the University of California in Davis in 2016. His research has primarily centered on the genomics of animal pathogenic fungi, and he's passionate about applying microbiome genomics to better understand the threat of pathogens posed to animals. And our other speaker is Dr. Lina Crespo. Dr. Crespo holds a DVM, a master, and a PhD from the Federal University of Rio Grande do Sul from Brazil. Her research focuses on assembling the health status of free-living amphibians in different agricultural environments. She's currently a clinical pathology resident at Tufts University with a focus on wildlife, parasitology, and infectious diseases. And with that, Lina, please take it away.
Thank you, Janina. Can you see my screen? Is everything working good? Yeah, great. So good afternoon, everyone. First of all, I want to say that I'm very excited to share this data with you and to thank MiDOG for the invitation. So today, the idea is to present two super interesting cases that come up over the course of a month. In late 2024 during our routine work at Tufts University. And first, I'll tell you about the cases, and then Dr. Yacoub will explain molecular analysis. And then finally, I'll discuss what we know about this organism. Okay, so everything started when this 7-year-old neuter male Labrador named Ryder was brought to the emergency service at our small animal hospital. So he was brought for evaluation of a one-day history of anorexia, vomiting, and lethargy. And he was bright and alert, but had mild hyperthermia, tachycardia, and he was fainting. So he had an external lesion that was an old small decline wound, and that was all. And then due to this increased respiratory effort, an ultrasound was performed, and it was found a small amount of pleural effusion and a moderate pulmonary consolidation in the right thorax. So naturally, the effusion was collected, and as you can see here, it had an incredibly high total nucleated cell count of almost 420,000 cells per microliter, and this is super, super high, something that we usually see associated with neoplastic effusions. But in this case, it was actually an exudate full of known to mildly degenerate neutrophils sometimes, and many macrophages. And every time we have an exudate, especially with such a high cell count, we need to look closely for bacteria. And in fact, we found a monomorphic population of gram-negative rods that were mainly associated with macrophages. But if you have ever seen a septic lesion or effusion, you know that we usually see bacteria being phagocytized by neutrophils. So again, finding them inside of macrophages was very unexpected. And we also have to consider that septic pleural effusions in small animals are typically associated with conditions that lead to a mixed bacterial infection. So penetrating trauma, foreign body, or even an extension from a pulmonary infection. So in this case, putting all these findings together made us think it could be something unusual. So this diffusion was submitted for bacterial culture, but unfortunately, despite all the supportive therapy, including treatment with ampicillin and enrofloxacin, our patient went into a progressive respiratory distress, and the owners elected for euthanasia the following day. So a necropsy was performed, and I'm sorry I didn't warn you people about the picture before, but it had this dark red diffusion in the pleural space, along with moderate diffuse edema and congestion in the lungs. So no potential in origin was found, such as esophageal or tracheal rupture or foreign body or even a penetrating chest wound was found. So on histopathology, as you can see here, the lungs showed a moderate diffuse pyogranulomatous pleuritis, but there was also mild focal fibrinous septointerstitial pneumonia. And I am just an aspiring clinical pathologist, but our anatomic pathologist who saw this case said this degree of inflammation made it unlikely that pneumonia was the initial cause. But something interesting is that no organisms were seen on routine H&E or even inspection stains. And we are going to talk about this later too. So then about a month later, we received this second patient, a 3-year-old neuter male Rottweiler named Caleb, that was presented for with a 100-day history of anorexia, lethargy, tremors, and dyspnea. So on physical examination, the patient was dull, had hyperthermia as well, tachycardia, and pale mucous membranes. So of course, with this non-specific clinical signs, we also performed diagnostic imaging, and then on ultrasound, there was a scant pleural effusion, and on radiographs, it was also seeing a soft tissue opacity in the left craniothorax. And then again, the pleural effusion was collected for analysis, and here you can see the main findings for this fluid. Once again, we do have a very high cell count, so this helps us to classify as an exudate. And one more time, we found this rods that show to be gram-negative on gram stain and mainly associated with macrophages. And there is something else super impressive in this fluid, which is this super high proportion of macrophages. The other one also had like 20-something%, but almost 70% of macrophages in a septic exudate is not something we see every day for sure. And then, of course, the sample was also submitted for bacterial culture, and I'll share the results for both cases in just a minute. But first, I just want to show you there was something extra in this case, which were this occasional atypical round cells. So these cells look very, very, very ugly, which naturally makes us think of cancer. But we need to consider this was a pyothorax, and also considering this degree of inflammation and the fact that the patient was a young dog of only 3 years. Cancer seemed highly unlikely in this situation. So at that time, we didn't know the significance of these atypical round cells, or whether we should interpret them as neoplastic or a reactive population, especially because some areas like these ones, we could see they looked to have a continuum between these large cells and macrophages. But unfortunately, this patient died in the same day. He suffered a cardiac arrest and did not survive despite trying cardiopulmonary resuscitation. And in this case, it happened so quickly that the clinicians didn't even have time to start antibiotics. And then again, a necropsy was performed, and that initial thoracic soft tissue opacity turned out to be a mediastinal mass. And it was confirmed by histopathology as an anaplastic large cell lymphoma. So this neoplastic process was also infiltrating multiple organs and was also accompanied by systemic histocytic inflammation. But as again, no bacteria were seen, even using these special things. So as I just mentioned to you, the clinical worsening in both patients was very, very, very rapid, and the culture results were not ready before their deaths. But you can see here that both cases yielded similar results, which were these pure colonies of known gram-negative rods, and no species identification was available to us. So these cultures were processed in an external lab. So we don't have the photos of the plates, but we did speak with one of the lab supervisors, and they told us that MALDI-TOF analysis indicated the closest match in both cases was Castellaniella defragrans. So this technique, MALDI-TOF, is a technique that uses laser to turn molecules into ions and then identify them using mass spectrometry. So this method is much more rapid and accurate than traditional techniques for bacterial identification, but you still need to compare the results with a reference database that is often more complete for well-known pathogens, and it needs to meet certain criteria, and these criteria were not met in this case. And then because of that, this information of the closest match was not included in the official report. And then with that, I will hand it over to Mark.
Hi, my name is Dr. Mark Yacoub, and I'll discuss the whole genome sequencing through MiDOG aspect of this case, which was an incredibly interesting story. So many of you might be familiar with MiDOG's all-in-one test, and if you're not, that's great. We offer broad coverage, high sensitivity and specificity, and DNA-based predictions of microbial pathogens, as well as rapid and detailed results, and it's effective in complex infections. So cases where you might have 2 or more pathogens in a single case, that's a great, we offer detailed results for those cases as well. And we can take it a step further with whole genome sequencing, which we're going to be officially offering in later this year, 2025, where we'll have parasite and DNA viral testing, virulence and bacterial toxin gene testing, and subspecies or strain level classification. We're still in the beta phase of that, but we are taking on exciting cases such as this one already. And if we move on to the next part of this slide here, just to compare these with PCR, PCR in comparison offers limited targets. So whenever you're designing a PCR primer set, you are making primers specific to a few or maybe 20 or 30 pathogens. So it's not that great for pathogen discovery if you have a case with an unknown species. And that's exactly what we had in this case. So on to the next slide, I'll introduce our MiDOG workflow. So this video here shows our process start to finish. So it starts off in the lab where we receive your samples, and those go through DNA extraction through first, these machines basically break up the tissue and release that great DNA from there that allows us to do our sequencing. Now, our whole process, start to finish, includes positive and negative controls to ensure accuracy of the results that we generate. Through here, we can show the flow cell, which is actually copying that DNA strand there to increase our chances of identifying your pathogen in these cases. And that strand is just duplicated here with its sister strand, and then the sequencing begins. And we get these A's, T's, G's, and C's, and that is your DNA sequence. We align that to the database that we've curated with over 64,000 bacterial species and over 10,000 fungal species to generate a library of target hits that we achieve through the sequencing. And just moving on here to Caleb, or case 2's report. So we found that Caleb's sample, or case 2, was dominated by an undescribed species of Castellaniella. You can see here it's given the SP moniker, indicating it's not a known or described species. Now this was fascinating because it was over 99% of the population and 6 million cells. But because it's an unknown species, it's difficult to know how this unknown microbe can cause disease. It's not been readily studied to know how these undescribed Castellaniella are infecting their hosts. However, next generation sequencing by MiDOG provides data beyond just identification. We can go a step further and look at the whole genome sequencing to further describe this novel bacteria. This is basically knowing the unknowns. We can take the unknown species and learn more about them. And so let's look into this bacteria's genome to find the answers we're looking for. Before I get into that, I want to backtrack a little bit and discuss what a genome is. A genome is the entire set of DNA, including genes, that a cell, or in this case a bacterial cell, contains. And why do we care about that from a clinical perspective? Well, the presence of certain genes can provide important context about a microbe's clinical relevance. Within a bacterial genome, there can be toxin genes that allow them to essentially poison the host or the patient, and antimicrobial resistance genes, or AMR, that allow them to resist treatment by drugs. And to get all these answers, we need to assemble the bacterial genome, and I liken this to assembling a puzzle. And here's how that puzzle works. Now, we take a large pool of large DNA strands, so this contains dog, bacteria, all of it, and we fragment those, so each strand is the same size. That way, each gets the same percentage chance of being sequenced. After fragmentation, we sequence those strands, and then essentially we put that puzzle together by pasting the ends onto each other by mapping ends, and then stand that strand out in the scaffolding strategy. Now, just a little anecdote about this case, which was actually pretty cool. Case 2, Caleb, was a male dog, and when I aligned the raw data, the DNA, to get rid of dog DNA so we could just focus on the bacterial, the dog genome I used was from a female. So the first pass of this, the Y chromosome was recovered. So that was just a funny anecdote, and really notes the importance of why it's important to indicate the or know the information about the patient, even from a genomics perspective. And here I show the genome of Castellaniella. This is just a circular genome map. It's a very pretty figure, but I want to indicate some important things that Castellaniella has that are not so pretty. So this genome contained a huge amount of self-defense functions. It had toxin genes, but it also importantly had toxin delivery genes. And why that's crucial is because not only to cause disease, a bacteria not only needs to make the toxin, it also needs to effectively deliver that to the patient to cause disease. And in this case, this Castellaniella species had both components. So it was able to not only make the toxin, but deliver them to the host. It also had a specific group of penicillin binding proteins, and these specific penicillin binding proteins indicate that this microbe has the potential to be resistant to carbapenems. So who is Caleb's Castellaniella species? As I mentioned, it's an undescribed species, but that doesn't mean we don't know anything about it. So what I'm showing here is the family tree of Castellaniella. All the labels here in black indicate the unknown species of Castellaniella. There are hundreds of unknown species of Castellaniella. They're not very well described. However, there are many known species as well. We can see here The N here represents the number of strains within each species. We can see here there are 4 strains of Castellaniella nitrificans known. There are 6 strains of Castellaniella defragrans and 46 of this ginsenoli. However, Caleb's Castellaniella here, which I show in red, doesn't belong to any of those known species or known groups of strains here. So who is this? Well, if we move on to the next slide here and zoom in a bit, we can show here that Caleb's Castellaniella is most closely related to this Castellaniella species that was isolated from a landfill in southern Ontario. However, those 2 species, Caleb's Castellaniella and the one from Ontario, are only about 93% genetically similar. Now that might sound like a lot. However, if you compare humans and chimps are about 95 genetically similar. And of course, we are very different species. And so this indicates that Caleb's Castellaniella is very different than all known and unknown species that are currently available. And so the question then is this a new species that became a pathogen? And the answer is likely yes. If we move on to the next slide, we can actually compare the results we see from case 1 on the left and case 2 on the right. Now for case 1, the species was identified as Castellaniella ginsenoli, and that was with Ryder. For case 2 with Caleb's case, we identified that as only Castellaniella sp until we dig further into the genome. And so just moving on to the next slide here, I just want to iterate that even though these species belong to the same genus, there is an ocean of genetic diversity that separates these 2 species. They are not even within the same clades or groups. And so from a clinical perspective, this indicates that both patients were infected with a very different bacterial pathogen, and likely indicates a different source of the infection as well. However, it also might suggest that Castellaniella are perhaps more important pathogens of animals than previously given credit for. And so just to summarize, our whole genome sequencing confirmed suspicions of a novel bacterial pathogen species of canines. And when we look within the genome and what parts it contains, we identify many toxin genes, stress response genes, and antimicrobial resistance genes. Together, these tell an important story. Castellaniella are known survivors. They are known to colonize and grow in very polluted man-made environments like landfills. And this makes them highly adaptive and resistant, and possibly even resistant to treatments in the clinical world. And through an unbiased and all-encompassing sequencing approach, we confirmed the presence of a novel Castellaniella species because we look for everything. If we were just relying on PCR, which struggles to identify novel pathogens, this case probably would not have been identified.
Cool. Thank you, Dr. Yacoub. Yeah, so these cases, they were very interesting for many reasons, but I think that before diving into the findings, I think it's important to give a bit more of the background, because at least for me, I had never heard of Castellaniella before these cases. So this genus belongs to the family Alcalagenaceae, which is characterized by gram-negative, facultative, anaerobic, malto, and short rods. And these organisms, they are classified as complete denitrifiers, and that means they can reduce nitrate to dinitrogen gas. And because of that, they are frequently found in nitrate-rich environments. So that makes them being highly adapted to human-modified environments, and they are often associated with contaminated soil and groundwater, agricultural systems, and wastewater. So now going back to the discussion related to this case, this is the first documented case of Castellaniella infection in domestic mammals. The only species from this genus previously linked to animal disease is Castellaniella ginsengisoli, the same one that was isolated in case 1, So this species was first described in 2009 during microbial screening of surface soil from a ginseng field in South Korea. And there are only 3 reports in the literature. One in an adult harbor porpoise found dead in the Netherlands, and 2 other they described mass mortality events. One of them involving Dorian picas and another with commercial poultry, and both were reported here in the US. So both of our patients presented with acute clinical signs, and they died shortly after admission. And we believe that several aspects support a direct association between Castellaniella infection and their deaths. And this include a severe inflammatory reaction against a monomorphic population of bacteria, the isolation of pure colonies from diffusion, and the identification of only one organism by next generation sequencing in each case. But of course, we need to keep in mind that the pathogenicity of Castellaniella remains poorly understood. So some of the authors from the other reports suggest they may act as opportunistic pathogens, affecting only immunocompromised animals. And the dog from case 2 did have disseminated large cell lymphoma, which would make sense, but the patient in case 1 had no obvious underlying disease. So taking that in account, along with the mass mortality event reported in other animals, Castellaniella may have a broader pathogenic potential beyond just opportunistic infections. And then I decided to give some extra attention to one of the papers because I think some people may feel a bit skeptical about the significance of describing one more bacterium among many well-known organisms, especially when it could be just a random opportunistic infection. So this is a report on a colony of Dorian picas, which are small, cute mammals from Mongolia that are related to rabbits that were brought to the Minnesota Zoo. So in their case, 16 animals died suddenly in the first 9 months, and many other animals died over the years, including both founders and their offspring. And then in 11 cases, they were able to diagnose a suppurative inflammatory disease associated with a bacterium that later was identified as Castellaniella, and most similar to ginseng isole, as I said before. So, and they couldn't identify in the other cases, sometimes because the carcasses were not available or something like this. So, at least in this case, we can say the bacterium had the capacity to spread through the animals in this colony and cause their death. And I think this is important to show since we only have a total of 2 dogs to show their findings, but at least in this report was something very, very impressive. So we don't know the transmission route for these organisms. So authors of the other reports have suggested maybe access through the alimentary tract, penetration wounds, ectoparasites, or maybe contaminated air or water. In case 1, the dog had a small dew claw wound, but it wasn't already healing, and the severity makes that association unlikely. And neither dog had a travel history outside the state, but both were regularly walked near ponds and lakes in Western Massachusetts. And although now we know they were from 2 different bacterial species, we looked for overlap between the hiking areas, and we didn't find any between the 2 patients. And they were also fed different commercial dry foods, and no other dogs in their households showed any abnormal clinical signs. And then I think that another important point to bring is and how this report highlighted the critical role of the clinical pathologist in interpreting samples. So in septic exudates, we typically expect to see larger numbers of neutrophils phagocytizing the bacteria. But in this case, finding the organisms primarily within macrophages suggests they may be capable of evading neutrophil killing and maybe survive inside of macrophages. And this points to a non-traditional bacterial etiology. And also, again, pyothorax in dogs is usually caused by mixed infections, whether from foreign bodies, penetrating injuries, or an underlying pulmonary disease. But we found no evidence of any of these in either case. And while bacteria were not seen in histopathology, we have to remember that histology has a limited resolution for microbes. And the same thing was also reported in the other case as well. So our conclusion here is that, well, this case report describes this bacterial genus that was previously unrecognized as pathogenic and now being implicated in fatal infection in dogs. And the identification was not possible using automated systems like MALDI-TOF, and in this case, it required next generation sequencing. But because of that, it revealed the potential novel Castellaniella species. And I think it's also fair to say that these organisms may possess a high pathogenic potential, and this should raise concern about the risks they may pose to both domestic and wild animal populations. And also, given their apparent preference for highly impacted anthropized environments, these findings could also serve as a warning, especially because their zoonotic potential remains unknown. And that's it. I would like to thank our collaborators once again, especially those at MiDOG, because you made it possible to sequence this genome and uncover these new findings. And I'm happy to take any questions you might have.
Thank you so much, Lina. That was wonderful. And thanks to you too, Mark. Yeah, we do actually have a couple of questions, but actually, since I can go first, Lina, you said this might be an emerging pathogen. So is this something that even the CDC is interested in, or is something that you think that should keep being monitored as we might have more cases emerging in the US?
Yeah, so this was something we discussed at the beginning after the first case we were able to sequence. And I honestly don't know. I think so, because it was so weird, the clinical presentation in both cases. We couldn't even treat the animals because of how fast they developed the clinical signs, and they got worse. We didn't get in touch with CDC yet. One of my supervisors was planning to do that. I think at least we should notify somehow, because again, we don't know. This is the first description in dogs, but I think there is nothing that avoid them to maybe be a problem to humans as well, or other species.
Mm-hmm. So in the chat, we have a question from Tamara Gull. She says, my lab has isolated a handful of Castellaniella fragrans from various specimen types, often in mixed culture with unknown clinical significance, but maybe 6 cases over the last 5 years. I think all cases came from Missouri. We have not isolated any non-defragrans Castellaniella, and we do sequence our unknowns that are thought to be significant. Okay.
Interesting. Thank you. Thanks for the information. Yeah, in this case, we had this pure colony in both cases, and only one bacterial morphology and cytology, and only one bacteria being sequenced on the NGS. So everything taken into account together makes us think it's only one bacterial species doing their job in here. But yeah, it's good to know, because so in one of the other reports, in the reports with a broiler chicken, they also reported different Castellaniella species being isolated with other bacterial species as well, and then will make sense to be just an environmental contaminant somehow. But in this case, it really feels it really appeared to be something of concern.
Yeah, and that's where it's so important to have a quantification aspect, not just present, absence, positive, negative, but actually have the cell counts to help determine if it's the main causative agent. Yeah. Dr. Sapanta is asking, have you identified any other novel genomes or have plans to investigate more in the future?
Hi, Dr. Zapanta. That's a great question. So as of now, we have at MiDOG sequenced and assembled 24 whole genomes of various parasites and pathogens of domestic and exotic animals. For context, the flatworm parasites such as tapeworms, MiDOG Animal Diagnostics has contributed 10% of all publicly available flatworm genomes to the NCBI reference genome database. So out of the 120, we've done over 10% of those ourselves.
Lina, do you think you would be using NGS testing more regularly in your lab because of cases like these?
Oh, absolutely. Yeah. Yeah, absolutely. Especially when we suspect it's something unusual and we cannot target something super specific that PCR requires. So yeah, that would be great.
Okay, it looks like those were all the questions that were in the chat. We have uploaded the CE certificate into the chat, so you can access it right there. There's also a QR code on the screen right now where you can access the website to download the certificate from there. And to all the attendees that are here present right now, we will also email you the PDF certificate following this webinar. So just to make sure you get all those credits. And with that, I would really like to thank everyone for joining us. We will have more webinar series coming this summer with all different kinds of interesting topics. And with that, just a quick reminder, MiDOG is an independent lab and we offer this type of diagnostic testing for all bacterial, all fungal, and parasitic and DNA viruses pathogens. Our results are typically ready in about 2 to 3 days. We are actively working on cutting down our turnaround time to just 1 to 2 days. So that will be implemented in probably about 3 to 4 months. With all of our samples, you do receive free shipping within the US. FedEx picks a sample up from your clinic, ships them overnight to our lab, and you get the reports in about 2 to 3 days, free with veterinary consultation. If you ever get a report back and you have any questions on how to interpret the results or what kind of treatment decisions to make for your patients, our veterinarians are available to go over a single report with you as well. And with that, I wish everyone a great rest of their day and I hope to see you again soon.
Thank you.